Buongiorno a tutti, è un piacere avervi qui. Vorrei ringraziare Ferdinando per aver organizzato qui questo meeting. Questo è il primo meeting che facciamo da 2 anni a questa parte, quindi speriamo davvero che sia di buon auspicio e che vi possiate godere appieno questa giornata, perché immagino che anche voi non abbiate partecipato in questo periodo a molte attività in presenza, per cui spero davvero che possiate passare una giornata bella, rilassante. Ho visto, rilassante, insomma interessante, perché ho visto che il programma di questo workshop, che è interessantissimo ma è anche molto fitto, quindi avrete davvero un sacco da fare oggi e vi auguro davvero che sia molto proficuo per tutti. Giusto 2 parole su questo dipartimento, così sapete insomma un pochino che cosa facciamo. È un dipartimento molto giovane, quindi è stato fondato, vedete, proprio alla fine del secolo scorso. Elevatissima multidisciplinarietà, quindi ci occupiamo di tantissime cose diverse e queste sono le afferenze. Siamo poi insomma tra tutti, tra PhD student, tutti circa 200. Queste sono le grandi aree tematiche di ricerca, quindi abbiamo l'area Health, Industrial Biotechnology, la Biodiversity and Food. Health si occupa proprio di malattie croniche, quindi l'argomento che discuterete oggi è proprio in tema con gli argomenti che facciamo in questo dipartimento. È stato qualificato il Dipartimento nel 2018 dal MIUR come Dipartimento di Eccellenza e ha anche ricevuto il finanziamento come Dipartimento di Eccellenza proprio per lo studio di malattie croniche multifattoriali, e quindi di nuovo il tema che affronterete oggi sulla biochimica dei tumori è proprio anche in tema con gli argomenti che abbiamo affrontato in questi anni grazie a questo finanziamento che abbiamo avuto dal MIUR. Quindi allora vi ringrazio tantissimo per essere qua, ringrazio Ferdinando e la Commissione per aver organizzato intanto questo meeting qua e poi per aver messo insieme questo programma molto interessante. E allora vi auguro una giornata, come dicevo, proficua e di interazioni importanti. Va bene, allora grazie a tutti e buona giornata.
Grazie, grazie Francesca. Continuo in italiano, cerco di prendere qualche altro minuto in italiano. Allora, tutto vi ringrazio di essere arrivati qua in questi tempi di Covid, anche se siamo alla fine, insomma non me l'aspettavo. Chiedo scusa a chi non ha potuto iscriversi, vi sembra che ci siano degli spazi vuoti, ma in realtà mancano ancora almeno un 40%, come vi dicevo. Filosofia del workshop. La filosofia del workshop è la scelta della scienza, non del passato, del presente. Quello che ha fatto la Commissione nel scegliere le persone, ma così come vedrete gli invited speaker non saranno presentati per la loro storia, ma solo perché quello che presenteranno in qualche maniera è interessante. Ed è quello che si chiede anche alle commissioni. Cioè le commissioni scegliete per la qualità della scienza e quindi troverete postdoc, giovani PhD che hanno qualcosa da raccontare. È anche un momento in cui possono crescere. Seconda cosa che volevo dirvi, è un workshop estremamente democratico. La linea che abbiamo tenuto è quella di far partecipare più persone possibili. Quindi degli 84 iscritti almeno 61 hanno un ruolo, che siano in commissione, che parlano, eccetera. È quello che abbiamo definito. Cioè diamo a queste occasioni la possibilità a giovani, meno giovani, di parlare, partecipare, crescere. E per noi diventa importante. Quindi mi raccomando, anche in futuro, se pensate di organizzare qualcosa, provate a mantenere una dinamica non del luogo, il luogo che si appropria dell'organizzazione, della gestione e tutto il resto. Organizzatevi, fatevi, come dire, servitori di chi viene, dando a tutti la possibilità di poter parlare. Terza cosa che voglio ricordarvi, tra un mese a marzo c'è l'Assemblea Generale della SIB e noi come gruppo dobbiamo presentare un programma annuale di attività. Quindi vi chiedo, se avete intenzione, avete in programma delle attività, contattatemi, perché possiamo provare un accordo per mettere un cappello, per far risultare che comunque biochimica dei tumori come gruppo si sta muovendo, che fa attività, che siamo in crescita, che riusciamo a coprire più punti. Se fate un workshop, se fate una giornata di studio ed altro, possiamo mettere il logo, provare a vedere delle sponsorizzazioni. Non siamo ricchi, perché vi dico, insomma, la SIB ci dà 1.000 euro, niente di particolare, insomma, però possiamo gestire qualcosa, e anche il fatto di avere il logo e di preparare durante l'anno una serie di eventi a livello nazionale, questo aiuta a farci crescere, perché alcuni di voi sono la prima volta che li vedo, cioè l'intento è quello di far avvicinare i giovani verso la biochimica. Per farlo dobbiamo trovare più momenti come questo. Va bene, io vi ringrazio ancora. At this point I think we have to switch to English. We have to start. The chairs, if they can come here. And I think we have one speaker that still is not here. Miglietta, last one. So, he's here. Okay, perfect. So, it's your time, it's your turn. Time. We have to be in time, you know? At 6 o'clock, start again the strike. So, we need to finish before, okay? Thank you.
I use this or? Okay.
Yeah, this is for... Okay.
Perfect. So, welcome. We start the first session of this workshop about metabolic rewiring cancer cells. So, the first speaker is Francesca Griesti from University of Foggia, and she will talk about unavailable metabolic plasticity of human osteosarcoma cells to improve cancer therapy.
Yeah, you want to?
Okay. Yeah.
That's it.
Okay. And the presentation. Okay. I have to close this one and open my presentation. Okay. Okay. This is the pointer. Just a minute. Okay. Okay. Good morning, everyone. It's a good pleasure for me to be here, opening this meeting and presenting our results about metabolic reprogramming in osteosarcoma cancer cell model. As all of us know, malignant tumors are metabolically heterogeneous since they display heterogeneous population experiencing various states of differentiation. Among them, cancer stem cells with a peculiar metabolic addiction became resistant to conventional therapy and thus leading to cancer relapse. In fact, while conventional therapies kill the bulk of tumor, cancer stem cell may remain viable and then later may reestablish the tumor, leading to relapse. So only if by destroying cancer stem cell, we are able to achieve a complete tumor eradication. Therefore, study cancer cell metabolism is no longer sufficient. We should analyze in detail what happens in cancer stem cells. But which metabolism should we target? So we look at the cancer and cancer stem cell metabolism, how it is modified by drug treatment, and how metabolic adaptation are affected by affected drug sensitivity. To this aim, we use the differentiated cancer cells MG63 and the cancer stem cells 3-BOS, whose metabolic phenotype well characterized by Chiaradonna group. And first of all, we confirm that 3-BOS show a more glycolytic and glucose-dependent metabolism compared to MG63 cells, which instead exhibit a glutamine-dependent metabolism, as already shown by Chiaradonna. So our question is, can we exploit these different metabolic features to sensitize cancer stem cells to chemotherapy? In other words, can we able to kill specifically cancer stem cells by the tailored manipulation of their energetic pathway? To answer this question, we look at the cell metabolism, drug responsiveness, and cell growth following specific substrate deprivation by using a complete medium, a glutamine-free medium, or a glucose-free medium. First, we look at the cell growth rate by phase contrast imaging and by real-time cell analysis, and then we evaluate cell metabolism by Seahorse technology. As expected, we observed a glutamine dependency for MG63 cells and confirmed the glucose dependency for 3-ABOS, as already shown by Chiaradonna. And then, by monitoring cell growth to the Xcelligence platform, we confirmed the greater sensitivity to a stepwise glucose reduction for 3-ABOS than MG63, which also in absence of glucose show a proliferation rate higher than the control. But by reducing glutamine concentration in cell culture medium, as expected, MG63 show a dose-dependent slowing down of cell growth, not detectable in 3-ABOS. Next, we look at the metabolic fluxes under similar condition, and as you can see, following glucose depletion, we confirm the oxidative and glycolytic glucose dependency only in 3-ABOS. Conversely, in response to glutamine deprivation, 3-ABOS are able to compensate a slightly decrease in basal OCR, enhancing glycolysis. This further confirms the glutamine dependence of MG63 for the metabolism and suggests also that cancer stem cell may possess a greater capacity of metabolic adaptation. So, to consolidate this observation and validate the reliance of the two cell models on distinctive metabolic pathway, we try to use the specific inhibitor of glycolysis 2DG and an inhibitor of glutaminase. And as you can see here, the inhibitor of glutaminase produces a significant arrest of the cell growth only in MG63 cells. On the contrary, treatment with the 2DG produces a dramatic arrest in cell growth for 3-ABOS, while MG63 cells stop to grow, but the viability is still preserved. So, putting together this first set of data, we confirm the reliance of MG63 cell on glutamine oxidation and of 3-ABOS on glycolysis for their survival and proliferation. So, our question is, we can exploit the specific substrate deprivation to sensitize cancer to the chemotherapy. In order to try to answer this question, we test the combination of low glutamine plus cisplatin at two different concentrations for MG63 cells, and the combination of low glucose plus cisplatin at the same concentration for 3-ABOS. Are the cells still resistant under this condition? So let's see MG63 cells first. As you can see here, at the higher glutamine concentration, cisplatin causes a dose-dependent inhibitory effect. And by reducing glutamine to a concentration still preserving viability, the proliferation rates were overlapping, and in particular at 5 micromolar cisplatin causes the same inhibitory effect observed at higher concentration. So this means that low glutamine sensitizes MG63 cells to cisplatin. However, it should be highlighted that these cells show a good sensitivity to the drug, unlikely the cancer stem cells. So let's see what happened in 3-ABOS. And as you can see here, at the higher concentration of glucose, the cancer stem cells are less sensitive to the drug, thus confirming the well-known resistance of these cells to the chemotherapy. And, but very surprisingly, in low glucose, the cells seems to grow even better. And this is more evident at the highest doses of cisplatin. So we can conclude that in low glucose, cisplatin induces a pro-survival effect in cancer stem cells, promoting cell proliferation. And this was very surprising for us. Next, we look at the cell cycle. And as expected, in a high glucose, it was observed a higher percentage of treated cells in G2M phase associated with a lower percentage in S phase. Very surprisingly, in low glucose, 3-ABOS cells show a delay in the exit from the S phase. So this higher persistent in S phase may account for cell ability to repair DNA lesion induced by cisplatin treatment and maybe to restart DNA synthesis, thus in this way exerting the survival effect we have already seen. Since it is well known that upon cisplatin, the cells undergo a massive DNA damage through the activation of p53, p21 axis, we further look at this pathway. And as you can see here, in spite of p53 activation, regardless of the glucose viability, surprisingly, the level of p21, a non-inhibitor of cell cycle, resulted to be down-regulated in low glucose. On the other hand, in high glucose, the canonical activation of this pathway was shown. So we suggest that all these findings could explain the similar distribution of the cell cycle shown in high and low glucose following cisplatin treatment. Moreover, cisplatin increases ROS production, and this is particularly evident in high glucose condition, likely due to the enhancement of mitochondrial basal respiration regardless of the glucose viability. On the other hand, glycolysis appear to be enhanced only in low glucose, and thus suggesting a mechanism by which the cells cope with the energy crisis due to the low of glucose, enhancing glycolysis. Next, CR's technology allows us to investigate the glucose, fatty acid, and glutamine dependence of these cells. And as you can see here, cisplatin increases the ability to use additional carbon sources, and this is particularly evident in the low glucose condition. In addition, cisplatin increases dependence on glucose, fatty acid, and glutamine, as you can see here. But if we go in detail, analyzing individually each substrate, we find that cisplatin didn't produce any variation in the ability to use glucose or fatty acid, but very surprisingly, it increases the glutamine dependency in those cells whose metabolic phenotype was more glycolytic, more glucose dependent, and apparently glutamine independent. This means that these cells increases their metabolic plasticity following cisplatin treatment. So, putting together this data, we can conclude that the enhancement of glycolysis, as well as the improvement of mitochondrial respiration and cancer stem cell plasticity, may provide the additional energy to sustain cell proliferation for a longer time in a low glucose medium. So all these metabolic changes may account for the pro-survival effect exerted by cisplatin in low glucose. Our take-home message is that the efficacy of specific metabolite starvation combined with the chemotherapeutic drugs depends on the cancer compartment, and thus suggests caution in using it as a generalizable curative strategy. What's next? Our basic research, our aim is understanding the mechanism underlying the effect of cisplatin in promoting cancer stem cell survival under specific metabolic condition. And if we have to image to transfer this knowledge into a clinical practice, we can image a two-step therapy in which the first one aims to kill the bulk of the cancer stem cell, of the cancer cells by conventional chemotherapy. And the second one addresses specifically against cancer stem cells by the fine-tuning of the nutrients and the tailored manipulation of the energetic pathway. Because have you just seen, the cancer stem cell became more and more resistant to both conventional and innovative therapies by continuously changing their metabolic features in response to the environmental modification. So the cancer cure seemed to be still far because the main obstacle remained the intratumor heterogeneity, that also means metabolic heterogeneity, mainly due to the higher plasticity of the cancer stem cell compartment. I would like to thank all the people involved in this project, and in particular my supervisors, Professoressa Claudia Piccoli and Professor Nazareno Capitanio, and all of you for the attention. Thank you.
Thank you, Francesca. Question from the audience? Okay.
Do you think this approach can be used also for testing other drugs? And in case, do you expect a similar behavior of these cells using other drugs and not only cisplatin?
Okay. Our data are about cisplatin, but if, as I told in the end of the presentation, I think that same problem will occur with other chemotherapeutic agent, since the tumor is show a higher heterogeneity, and the cancer stem cell compartment with the metabolic plasticity, also in the case of other drug, could show the same paradoxical effect. So that's, I think, yes.
Thank you for your presentation. My point is that if you think the tumors, you have two different compartments. One is that it's very toxic. You can imagine that in this central part there is more glucose, and the outside you have glucose and so on. You can imagine that cancer stem cells may be preferentially in one of these two compartments. When you treat with the cisplatin, you can increase survival of stem cells in the hypoxic area or in the external part. Do you know something about this kind of treatment?
We don't look at this in particular, but what we can image is that behind the compartmentalization of the cells, what happened with the cisplatin is that in low glucose, due to the, I don't show the results, but we look at the inner membrane potential, and we saw that in low glucose there was a reduction of this potential, and this may account for a reduced uptake of the cisplatin inside the cells. So we postulate that this is the reason of why in this way could be occur the ROS-dependent damage induced by cisplatin, as I showed you before, and consequently in this way the cells are able to escape to one side the DNA damage, as I show you in the cell cycle analysis, and from the other side the ROS scavenger system does escape the ROS damage induced by cisplatin that also occur in the DNA. So I think that this will be a reason of this effect.
So did you check, so you showed that there is this accumulation in S phase. Did you check for checkpoint?
Yes.
For in this
yes,
phase,
I have the results
Yeah,
in the end.
they are
Sorry,
still.
unfortunately, we don't have
Just
time.
a second.
for-
Here we go.
We are
We
late.
look at the other checkpoint. So
No, I was thinking about check 1, check 2, because this is flooding is DNA damage. If you check for this, okay.
Okay, thank you so much. We need to jump to the second presentation from Roberta Rapuano from University of Sannio, entitled Cladrоспорol negatively regulate 3T3L1 cell differentiation and through adipocytokines synthesis and secretion influence HT29 colon carcinoma cell proliferation.
Good morning. Thanks for the presentation. I performed this experimental approach thanks to my supervisor, Professor Angelo Lupo, and we focused on the role of these metabolites, cladosporols, in metabolic reprogramming in tumoral cells. So, cell-to-cell and organ-to-organ crosstalk is a feature of the most evolutionary developed organisms in determining functions in normal cells and dysfunctions in pathological cells. So to break these interactions is really important, I think, to avoid, for example, the insurgence of several pathologies, as for example, cancer. In fact, if we imagine at the tumoral microenvironment, we have, as we previously said, a really heterogeneous population. In fact, the surrounding cells of the surrounding tissues is, for example, fibroblasts or adipocytes or endothelial cells. They present an alteration in their cell biology. So it is now considered important to understand, for example, the reciprocal interaction between the stroma and the tumors to understand how, for example, the adipose tissue can nurse the tumors. So let's imagine to the formation of two compartments in which the tumor displays the role of a parasite by sequestering all the molecules, as for example glutamine or lactate or fatty acids, by activating catabolic pathways, and they use these molecules as substrate for the activation of metabolic pathways and to nurse the tumors. In this scenario, for example, we focused on the role of adipocytokines, precisely on the role of adipokines leptin and adiponectin, because we wanted to analyze the mechanisms underlying these interactions, for example, understand better the links between pathologies as obesity or cancer. For example, in our laboratory, we are focusing on the role of colon carcinoma cancer and prostate cancer. This is the first aspect we wanted to analyze, the natural molecules. We focused on the role of cladosporols that are secondary metabolites derived from Cladosporium tenuissimum. They have been previously studied in colorectal cancer, and when they bind to the pocket of their receptor PPAR gamma, they have several actions, as for example mechanisms. They can control the proliferation of colorectal cancer cell, and they can also modulate adipocyte differentiation in our approach, for example, in 3T3L1 fibroblast preadipocytes. Okay, so we are talking about differentiation. Here, just one moment. Okay, here, we analyzed the role of cladosporols in differentiation. We performed the differentiation using the 3T3L1 preadipocytes. We differentiated them using a mixture, MDI, composed by insulin, desamethason, and EBMX. And then we used these two isoforms of cladosporol, cladosporol A and cladosporol B. Here, we stained them with red O, and there is a reduction in lipid droplet accumulation compared to rosiclitazone that is a well-known agonist of the receptor PPAR gamma, and to the all differentiated cells. So to control, we analyzed the nuclei by hematoxylin staining, and then we give you a photograph of the plates in which we induced this differentiation. We evaluated this staining, and we analyzed through spectrophotometric assay. And as we can see here, also it is reported that there is a reduction in droplet accumulation after using cladosporol A and cladosporol B. So to better understand the role of these compounds and these receptors, we wanted to analyze as first the adipogenesis related genes. As first, PPAR gamma. PPAR gamma is one of the master regulatory genes of adipogenesis. So after treating cells with cladosporol A and cladosporol B, there is a reduction in adipogenesis. And all these genes that we analyzed are target genes of PPAR gamma. Also them, early and late adipogenesis-related genes result to be decreased in our real-time PCR analysis. We wanted to confirm this by analyzing also the protein levels of these markers. And also here, there is a reduction in protein expression after treating cells with cladosporol A and cladosporol B. To better understand the role and that these effects were due to the real binding of cladosporols to PPAR gamma, we treated cells with a specific inhibitor of the receptor GW9662, because we wanted to occupy the pocket in which the cladosporols should bind, but there is GW, and so they cannot bind. And so we repeated, we repeated the oil red O staining, and in all treated cells with cladosporol A and cladosporol B, and in all pre-treated cells with GW, there is a reduction in droplet accumulation that is shown in coloration, in staining, in plates, and also in absorbance. So we wanted to focus on the transcriptional activity. So we performed a luciferase assay in which we transfected this sequence, PPRE, and we analyzed as first the transcriptional activity after treating cells with cladosporol A and cladosporol B in pre-adipocytes, and then in mature adipocytes. There is a reduction in transcriptional activity both in pre-adipocytes and adipocytes mature. Then we pre-treated cells and we did the same analysis, and there is a reduction also when pre-treat cells with GW9662. So we confirmed all this by analyzing the expression level of PPAR gamma. What happens? Here we analyzed both the isoforms of PPAR gamma 1 and 2, in which the second isoform is an isoform containing 30 additional amino acids and terminus, and there is a reduction also in expression. So we confirmed this data. As I previously said, we wanted to analyze the interactions between adipose tissues and the tumoral tissues. So we focused on the role of secreted factors, and we used as first trititrial-1 pre-adipocytes, as I have previously shown. We differentiated cells, and then we collected all the medium from these adipocytes, and we used this to cultivate sbaglio sempre. Un attimo. Okay. We used to cultivate HT29 cells. As first, we evaluated the proliferation of these cells HT29 after 48 and 72 hours of conditionment treatment. And then we performed a wound healing assay by using this media, and we said that in HT29 treated with conditioned medium from cladosporol A and cladosporol B, there is an inhibition in migration. So there is an inhibition in proliferation and in migration. But someone can ask me, but there are cladosporols in the medium, so probably the effect is due to the presence of these drugs in the medium. So we repeated the experiment by removing all the drugs for the last 2 days, and the result was unchanged. So we are showing you here that also by removing the cladosporols, there is an inhibition in migration. We further proved them by analyzing beta-catenin level by Western blotting. There is a reduction in beta-catenin levels by treating cells with conditioned medium from cladosporol A and cladosporol B. There is a reduction in cyclin D1 and an increase in P21 expression level, indicating that there is a cell cycle arrest. And at least we analyzed the precursor of caspase 3, and the decrease in the level indicates that we are probably in presence of apoptosis. But I previously said that we were focusing on the secreted factors, and we saw that the results previously shown were due probably to the presence of secreted factors by the adipocytes and not to the presence of cladosporols. So here we are showing you the intracellular and extracellular level of 2 adipocytokines of relevant interest that are adiponectin and leptin. The intracellular level of leptin is reduced after treating cells with cladosporol A and B, and there is also reduced secretion in the medium. In adiponectin levels, there is a reduction in intracellular level, but there is an increase in secretion of these adipokines. So here I'm showing you a little scheme of all the things that I have you explained until now. So, in conclusion, our result can support our hypothesis. So cladosporol can block the proliferation of colorectal cancer cells, as we previously said. Maybe they can also inhibit this interaction between the adipose tissues and the progression of tumors. And so for us, this is a good approach. Thanks.
We have now time for a couple of questions.
Thanks for your talk. Treated cells produce a lot of cytokines or growth factors.
Sì,
You
yes.
check if your drugs affect these molecules. So if your drug reduces the production of cytokine or other growth factors that can affect the...
Okay. Now we are focusing only on adipokines and leptin. But we wanted also to analyze not only, for example, the adipogenesis, but also, for example, the adipogenesis. So to understand these mechanisms, for example, to analyze how the adipose tissues can influence the tumoral factors, but we wanted also to understand if there was a change by inverting this process. For example, what happens if we treat HT29 cells with cladosporols, and what happens to all the adipocytokines? This is a question mark that we are trying to do this in the future. Thanks.
Okay, thank you so much. So we move on. Silvia Trombetti, please. She will talk about dysregulated expression of the transcriptional factor GATA1 rewards metabolic activity in myeloid leukemia cells.
Good morning to everyone. Thank you for the presentation. And I will talk about the transcriptional factor GATA1 and its isoforms, how they are involved in metabolic rewiring. GATA1 is a key hematopoietic transcription factor involved in differentiation and proliferation of myeloid precursors. GATA1 encodes a DNA binding protein that contains 2 zinc finger domains and 2 transactivation domains, the N-terminal and C-terminal domain. The GATA1 short isoform lacks the N-terminal domain, and it is involved in an assembly of different transcriptional complexes. The overexpression of GATA1 short isoform the hematopoietic disorders are characterized by the overexpression of GATA1 short isoform and thus promoting cell proliferation. These disorders are also characterized by the alterated homeostasis of reactive oxygen species. And given the potential leuchemogenic role of GATA1 short as well as stress conditions, we wanted to verify if there was a correlation between the expression levels of GATA1 isoforms and different redox states, cellular redox states. The evaluation of ROS levels in K562 cells transfected with the 2 isoform of GATA1 showed increased cytoplasmatic ROS levels in GATA1 full length, while we found a marked increase of mitochondrial superoxide only in GATA1 short cells. However, when we normalized the mitochondrial superoxide on mitochondrial mass, we found a reduced ratio of in GATA1 short cells, thus indicate a lower mitochondrial superoxide concentration in GATA1 short cells. We next asked whether this increased in mitochondrial mass was related to changes in number or size of mitochondria, but we found no variations in mitochondrial DNA content in GATA1 short cells. On the other hand, we evaluated the expression levels of proteins involved in fission and fusion processes, and we found that GATA1 short correlates with the overexpression of mitofusin 2. This condition highlights that the increased mitochondrial mass is related to enhanced fusion processes in these cells. It's well established that fusion helps to mitigate the stress condition by mixing the content of partially damaged mitochondria as a form of complementation. So we hypothesized that mitochondrial metabolism is impaired in these cells. To verify this hypothesis, we analyzed the mitochondrial metabolism using sears, and we found that mitochondria in GATA1 short cells showed reduced basal and maximal respiration, reduced protein leak at pre-production, and also a lower respiratory capacity. This result was indicative that in GATA1 short cells showed limited oxidative phosphorylation efficiency. Mitochondrial superoxide is generated mainly at complex 1 and complex 3 of electron transport chain. However, recent data showed that also dysfunction of complex 2 can generate high levels of mitochondrial superoxide. These alterations may be due either to the overexpression of subunit of complex 2 or to point mutation or to splicing variants. All these alterations can cause a disassembly of complex 2 that is accompanied with a reduction in SQR activity. Data previously obtained in my laboratory showed a different redox state of Hpv moiety in these cells. And on the basis of all these observations, we wanted to test the SQR activity in our cells. And interestingly, we found a reduction of this activity only in GATA1 short cells. To better understand if this reduction was correlated with the alterations in the structure of complex 2, We next evaluated the expression levels of all the subunits of complex 2, and we found higher levels of SDHC only in GATA1 short cells. 2 splicing isoforms have been described so far of SDHC, lacking the exon 3, in which there was a SQR activity region, and exon 5 that is linked to heme binding region. The 2 isoforms are called delta 3 and delta 5 respectively. We thus evaluate the expression levels of these isoforms, and we found that along with the full length, also the 2 splicing variants were found elevated in GATA1 short cells. To better clarify that GATA1 short directly alters the SDHC levels, we next silenced endogenous GATA1 short, and we found that the silencing of GATA1 short is correlated with a reduction of total and in total expression levels of SDHC, and also a reduction of its isoforms. Therefore, abnormal levels of mitochondrial superoxide associated with the GATA1 short can be related in part to complex 2 disassembly and its impaired activity. As a whole, GATA1 is able to influence the morphology and the metabolic activity of mitochondria. More recently, to better clarify, to better investigate this issue, we performed metabolomic and lipidomic analysis. And recent data obtained so far showed in GATA1 short cells a reduction in mitochondrial citrate content that is related to higher levels of citrate lyase expression. We also found in GATA1 short cells, higher glutamine and glutamate content that well correlates with the glutaminase expression levels that was found increased in these cells. This result showed enhanced glutamine metabolism in these cells. Therefore, Sears analysis also showed in GATA1 short cells elevated extracellular acidification rate, thus indicating an enhanced glycolytic flux in GATA1 short cells. This result was also confirmed by the overexpression of HIF1 alpha in these cells. All these data, all these findings recall the phenotype of leukemia cells. The lipidomic analysis has showed so far an alterated pattern of sphingolipids metabolism with reduction in specific ceramides species that is in agreement with the higher expression level of the specific alkaline ceramidase 2 that specifically catabolize these ceramide species. Even in this case, the overexpression of these ceramidase, as well the alterated sphingolipid metabolism, recall to phenotype leukemia cells. Finally, we confirmed our results also in bone marrow samples of myeloid leukemia patients. And we found, in fact, that the diagnosis higher levels of GATA1 short that then normalized the remission, and similar trend also for SDHC levels and HIF1 alpha. In conclusion, the short isoform of GATA1 leads to different ROS production and compartmentalization, promotes cellular energy metabolism rewriting through mitochondrial remodeling, and is associated with the sphingolipid metabolic rewriting. These results contribute to elucidate the molecular pathway through which GATA1 short exerts a proleukemic role, and we believe that this study identifies a new potential molecular target for the development of more effective therapies in myeloid leukemia. Thanks to all of you and to my professor.
Question from the audience?
Very nice presentation. I have a couple of questions. The first one, in order to define the morphology of mitochondria, have you considered to analyze them with the confocal microscopy? And in addition, other markers like OPA1 and the phosphorylation of DRP1?
Okay, thank you. We made experiment with the confocal microscopy, but and we show it's not really easy to understand the morphology of these mitochondria because the K562 cells have a really big nucleus and really little mitochondria that are not so easy to study with the microscopy. And we also we tried to evaluate the expression levels of OPA1, but our antibody was not going yet. And we will also try to see the expression levels of the DRP1 phosphorylated.
And if I can do another question? Yes, very quickly. It could be interesting to study the role of TRAP1. Do you know TRAP1, which is a very known inhibitor of succinate dehydrogenase? Have you tried to test it?
We didn't try, but of course is in our future perspective also because we would like to change our model, trying to see also with the microscopy the change in morphology of mitochondria. Thank you.
Sphingolipid metabolic unbalance and the mitochondrial dysfunction which you start. And then I have also another question. Did you check also other proteins, I mean of inner membrane mitochondrial proteins or matrix beyond the one that are involved in the fusion fission? Because the only mitochondrial content that you analyzed, I understood, was mitochondrial DNA, that it didn't change.
Yes,
yes. If you analyze other mitochondrial, mitochondrial protein in any way, to see what's going on.
We didn't check other mitochondrial proteins.
And for the sphingolipid metabolism?
And because, as you know, the ceramides are important molecules involved in cell proliferation and apoptosis resistance. And in our previous data showed that GATA1 short cells have under pro-apoptotic stimuli are more resistant to the GATA1 short cells. So we think that the sphingolipids and also the content of other lipids in the membrane could be involved in the signaling related to apoptosis resistance and cell proliferation. But they are only preliminary data. We are just starting to investigate the lipid content.
Ceramides are also pro-apoptotic in some conditions, but
yeah.
I have not clear the level of changes of specific ceramides, probably.
Yes.
I missed it.
We found that these specific ceramides were reduced.
Were reduced,
yeah.
So that's why there is it.
Yeah.
Okay, sorry.
That
Thank you.
Can show apoptosis resistance phenotype. Thank you.
Thank you. We have our last presentation before coffee break from Stefano Miglietta from University of Bologna, entitled MCJ DNA JC15 Mitochondrial Chaperone as a metabolic balance needle in ovarian cancer chemoresistance.
Okay, thank you. Bravo? Okay. Okay, thank you for the presentation. I have to open mine. Okay. Today, good morning everyone. Today I'm presenting my research project which concerned the study of the role of MCJ, cochaperone protein, mitochondrial protein, as a metabolic balance needle in ovarian cancer chemoresistance. Ovarian cancer is a low incidence gynecological cancer. This is extremely invasive with high lethality. The high mortality is due to, because the ovarian cancer in the early stages is completely asymptomatic. And when the first common symptoms appear, the ovarian cancer is already spread into the abdomen and create the abdominal carcinosis and reach distant tissue sites and metastasize and develops chemoresistance to conventional therapy, cisplatin-based. Chemotherapy is a cogent aspect of ovarian cancer. In particular, recently it has been demonstrated that chemoresistance to conventional therapy is strictly related to the metabolic capacity of the tumor in order to adapt to the tumor microenvironment. Chemoresistance for instance, chemoresistant ovarian cancer cells are demonstrated as more dependent on glycolytic metabolism rather than the oxidative phosphorylation. In this context, it's interesting what has been reported about MCJ, mitochondrial chaperone. There is a cochaperone protein in the mitochondrial membrane that is encoded by the gene DNA GC15. MCJ has been shown as involved in the mitochondrial folding and import of protein into mitochondria mediated by TIM23 complex. And then it's also an interactor of the mitochondrial permeability transition pore. But what we know in literature about the functions of MCJ, the knowledge is very low. But what we know is in a non-cancer context that MCJ seems to be able to modulate the assembly, the activity of the mitochondrial supercomplexes. But it's interesting what we know in cancer for the reason that MCJ seems to be deregulated with a lower level or absent in a lot of type of cancers, and in particular in ovarian cancer, where MCJ, the regulation, is related to the chemoresistance cisplatin-based onset. Recently, it has been demonstrated that MCJ in breast cancer is able to restore doxorubicin sensitivity with its impact on the mitochondrial bioenergetics. For this, in this frame, we try interesting to investigate in MCJ as a modulator of ovarian cancer drug resistance occurrence and as a modulator of mitochondrial bioenergetics, in particular studying MCJ as a modulator of the bioenergetics and the metabolism, and by studying its involvement in the mechanisms underlying ovarian cancer chemoresistance. For this purpose, we analyzed the expression of MCJ in a panel of cell lines, and we found that MCJ is completely absent in the two cell lines, A2786 cell line, that is a cisplatin-resistant ovarian cancer cell, and HK2 cell lines, that is a non-cancer renal cell lines. So we try to overexpress MCJ in both cell models in order to see what happens in bioenergetic of mitochondrial distal lines. We found that MCJ in both cell lines is able to increase the oxygen consumption rate capacity at both basal level and uncoupled maximal OCR. And accompanied to this profile, also we saw a boost effect in ATP production in both mitochondrial and glycolytic ATP enrichments. This high energetic profile is associated also with an increased specific redox activity of mitochondrial electron transport chain activity that is significantly higher in both cell models with the MCJ overexpression. This high energetic profile induced by MCJ is strong and it's encouraging because we found a positive and strong and also a significant correlation between the MCJ expression and the maximal bioenergetic potential in a panel of cell lines. We know from the literature that an induction of mitochondrial activity can induce the production of reactive species and in turn oxidative stress that could induce, for example, lipid peroxidation of the membranes and then produce the reactive products such as 4-hydroxynonenal that can perform adduction on protein and then reactive protein. So we know now there is a technique to recognize 4-hydroxynonenal such as a direct correlation of the lipid peroxidation level. And what we interestingly found is that MCJ seems to be able to increase lipid peroxidation in both cell models, suggesting an impact also in oxidative stress. A brief summary of the part, MCJ seems to be an enhancer of mitochondrial oxygen consumption and ATP production, and this profile is accompanied with an increase in electron transport chain activity, and that could be related to cellular redox imbalance. But what we know from the literature is also that MCJ, when in ovarian cancer in particular, when the ovarian cancer become more resistant to conventional platinum-based therapy, we saw the regulation of MCJ. In particular, we found by analyzing the expression of MCJ in our cell models that the resistant cell line has an absent or lower expression of MCJ compared to the chemosensitive counterpart. So we tried to overexpress MCJ in both cell lines, and in order to see what happened in the response to cisplatin-based therapy. We found that in increasing concentration of cisplatin, MCJ is able to reduce the viability of both cell lines, and this is accompanied with a strong reduction of the IC50 of the drug for both cell models. In order to see if the sensibilization to cisplatin is accompanied with a reduced parameter or tumorogenic parameter, we try to perform a chronogenic assay in untreated, treated with cisplatin conditions for both cell models. So what we found is that MCJ is able to reduce in both conditions the number of colonies and the colonies protein contact in a 2780 cis cell line, but only the colonies protein content in cis cell lines. According with this, we investigated also in the migration, proliferation, and invasion profile of these cell models. In particular, in A27 cis cell lines, we saw that MCJ is able to reduce the migration ability, as we can see in the wound healing assay, and to reduce the proliferation, given the fact that the overexpressing cell lines have an higher doubling time. We found also by evaluating the ability to the cell model to form the buds for the invasion in 3D spheroids, that MCJ strongly decreased the ability of these cell lines to perform invasion. But how MCJ could, mitochondrial protein, could regulate ovarian cancer proliferation, migration, and invasion? Among all the pathways that are activated in ovarian cancer that can be influenced by oxidative stress, for example, we focused on beta-catenin pathway that has been reported also to be negatively regulated by oxidative stress. When it's active, beta-catenin is free to translocate to the nucleus and transcribe gene that codify for proteins involved in epithelial transition, mesenchymal transition, multidrug resistance, and cell proliferation. Then we checked the signaling of beta-catenin in our cell models, and we found that beta-catenin is downregulated when MCJ is overexpressed, and also all the downstream targets that are mesenchymal-like targets like SNI1 and catenin, MBCG2, are significantly downregulated, probably suggesting that MCJ is able to reduce the mesenchymal phenotype of these cell lines. This aspect is observable also by eye, because in our cell line that normally is composed by a mixed population of rounded-like epithelial cells and elongated mesenchymal-like cells, when MCJ is overexpressed, the mesenchymal-like population tends to lose, so mirroring what is happened at the molecular level. So by concluding, we saw that MCJ is able to trigger mitochondrial respiration in a no-cancer context and ovarian cancer cell lines, and seems to be strongly involved in the mechanisms of chemoresistance and ovarian cancer progression. In particular, seems to have an anti-tumorigenic-like activity, since MCJ, by impact on the ATC activity, could produce an oxidative stress that in turn block the beta-catenin signaling axis. So reducing the mesenchymal phenotype, the resistant therapy phenotype of the cisplatin chemoresistance cancer cells, in order to render them more similar to the chemosensitive one. I would like to thank my mentor, Professor Anna Maria Porcelli, and also Professor Anna Maria Ghelli and Professor Giuseppe Gasparra, all my colleagues and friends of the Mitochondrial BNB lab and Medical Genetics lab, all the funding, and all of you for your attention.
Thank you. There are any questions from the audience?
It's very simple. Have you tried to test ferroptosis?
No.
Ferroptosis is, it could be, it could fit very well in your data, because at least from what I saw, because it is a hydrodependent oxidative cell death, which causes the peroxidation of lipids and has an effect on the metabolism of the cell. So I know it is quite difficult to test ferroptosis, but it could be very interesting in your system.
Thank you very much. Yes, because we are thinking about, we are now working on this aspect to evaluate if it's something induced by ROS or NOS level. We don't know which type of oxidative stress, because also ovarian cancer cell lines have higher level of antioxidant enzymes, so it's very difficult. For example, we were wondering that MCJ is an inductor also of the lipid droplets formation, and we are, for example, hypothesizing that it could be a buffer mechanism to buffer all the oxidative stress, the induction of lipid droplet formation. But it's an interesting point. Thank you.
It's very interesting, this, what I missed probably because I don't know. What about the function of, if it is known about MCJ in normal cells, and if you know where is it compartmentalized into mitochondria, or it's...
I saw in the introduction briefly, but it's, yes, it's an integral protein of the inner mitochondrial membrane, and normally it's the simply co-chaperone protein of the TIM23 import complex. So, it's an interactor of MAGMAS and mortalin, and for this reason is also involved. It's an interactor of the mitochondrial permeability complex.
But this does it mean that also in normal cells there is this increase in biogenesis and bioenergetics?
Yeah, probably, yes.
So this could be a shortcoming of any use of this kind of a molecule for treatment. I mean, this
Yeah,
could be a problem, I think, because anyway it's affecting also normal cells in a similar way.
But we don't know. For MCJ, the knowledge is very, very, very low in literature. Only this year came out just 2 papers that describe MCJ as a target, in, for example, in cancer context. But in general, it's studied as a protein, is a physiological protein of the mitochondria.
That was my questions, wondering. Okay.
Thank you.
Okay, thank you. If you,
Thank you.
If we don't have any other question, we can go to our coffee break time. And just a few words to invite you to come back in time at 11:30. Thank you so much.
So let's start again. So it's a great pleasure for me to present here Susanna Chiocca. We don't need really to talk about what she did in the past, as I said before, but what she is going to say this morning. It's quite a little bit different from the typical talk that we usually listen in this type of the congress and seminars, but I think it's quite interesting also this year that we talk about woman, woman, woman. And here we are discussing a little bit on sex and gender, so the difference. So the title of talk, let's start, is Sex and Gender Dimension in Science and Research. Perfect. So let's see what she would say. Thank you.
Here we are. So thank you for this invitation to talk about a theme that is so dear in my heart.
The microphone, please. Please, the microphone. We need the microphone.
The ice cream. The ice cream.
The ice cream.
So the good thing is I've been giving this talk more and more this last year online, which is good. It means that the issue is becoming more, becoming more aware of the issue. And this has got nothing to do with gender quota, men and women. It's something more profound that starts, of course, with the fact that so many times scientists don't see things simply because, you know, we think that we're all the same, and so mistakes are made throughout. So this idea was long in my heart, in our heart in the lab, but then we got funding from the European Commission in 2015, and you know, when you get funding, then you can develop your ideas. So what I'll tell you today is I'll briefly just mention about the project. It's important. And then I'll tell you what it means, why do these terms, sex and gender, matter in science, give you some definitions, example, etc. And then I'll briefly touch upon, I don't have enough time, but we've built a teaching module that's open to everybody in case anybody's interested in taking it. So about Libra, just quickly, Libra was stemmed within EU Life, which is an alliance of institutes in Europe. And we have, you know, we get together, we write grants, and so it was stemmed from the CRG in Barcelona, and this was all the institutes that belonged to Libra. And we had a nice committee, advisory committee. These are all gender experts in Europe. So for example, Kurt Rice is the rector of Oslo University, Ineke Horg and Ines, they're all experts in gender and science matters. So how did I get interested? So I've been working on viruses basically since my PhD, and I love many themes. And most recently, in the last 20 years, I've become interested in looking at HPV related diseases. And when I first started working on this, you know, I realized, okay, well, you know, HPV hits everybody, pretty much, men and women. And of course, it's organ dependent. So depending on the organ, you see many men. And of course, that's the case because the virus is sexually transmitted. So you do expect to have all sexes become infected. Nevertheless, it was more always considered as a woman issue. And that changed recently. Why? Because of the virus gaining momentum because of the rise of oropharyngeal cancers, HPV-induced oropharyngeal cancer, especially in the male population. So that triggered a bell in my head, and we went back and we looked at what happened and what we call the genderization of HPV vaccines. So back then, when the vaccines were first approved, these were advertisements. And you can clearly see the advertisement was clearly hitting the women, the girls. So there was immediately a stigma on this, and it was wrong. Now it's changed. Of course, the vaccine is also developed for boys and for men, but nevertheless, at the beginning, it was always, well, okay, girls and women need to be vaccinated, and that was wrong. That was the wrong way to look at this problem. So at the time, I had a PhD student in the lab, Paolo Maugeri, who was a philosopher as well. We started developing the idea of what can we do to change this a little bit, to make, our minds more open. And so again, we got Libra. And Libra basically worked on these 4 big areas of intervention. And I won't talk about these, but they're important now because, as you know, to get public funding, you need to have your gender equality plans in place. All institutes to get public funding. As of 2022, for example, European Commission, Regione Lombardia, Cariplo is going to start. You have to have these plans. And part of these plans also include the idea of how to integrate this sex and gender dimension in science. So we built a lot of training material. They're all open. They're on a website. And I will briefly go through how do we get to this. So first of all, let's go through some definitions, okay? So definitions are important. We always often use these 2 terms interchangeably, and wrongly so, because when we talk about sex, we should only refer to biology. So characteristics in terms of organs, function based on complement physiology. So sex is really understood as living beings, be either male or females or intersexed. Males and females, so we have about 50% in the world, women, males, 50% female. And intersex, there's about 1 to 2%. There's not much well studied yet, but hopefully it's coming along. Gender is completely different. This is a social cultural process, and it refers really to attitudes and values that sanction the feminine and masculine behavior. And it's important because this process can affect technology, environment, and knowledge. So, to get into the idea, so gender, we have to think about gender norms, so the attitudes and the behaviors that we think are more appropriate for women, men, and gender diverse individuals, the attitudes that we have, that they're constantly reproduced through social institutions, in families, schools, workplaces, text, movies, everything we are full with these gender norms. And it's important to understand them because they influence the development of products and technology. And they draw upon and they reinforce gender stereotypes, which are these widely held beliefs that we have about women, men, gender diverse individuals, what's feminine and what's masculine. And they're constantly in flux, meaning that, the way you behave in the 90s is different from the way that people behaved in the 90s is different from the way they behave now. The way people, even in Italy, north, south, in Europe, Germany versus Italy, and America, so they're constantly in flux. Also, the way you behave at home is different from the way you behave at work, so it's a continuous flux. And I recommend you to look at this website that was developed by Londa Shibiberg, who is a historian at Stanford University. And then, of course, we have the famous gender stereotypes that we have, all of us have them every day. They're the widely held beliefs of what we think, are men, women, what they should be, masculinities and femininities. So why is it important in science? It's important in science because when we think of a scientific problem, we have to question, what does it really mean? And if we are addressing the evolving needs and the roles of women, men, and gender diverse people. So we are in an era now where it's the end of a one-size-fits-all, right? We're in an area of personalized medicine, precision medicine, And of course, we have to think of making research as innovation more responsible to the different needs of all specific individuals. And the perfect example of this is the way seatbelts were made back years and years ago. The crash dummies that were used were based upon the 70-kilogram white male, so the statue and everything. So of course, at the beginning, what happened is that the seatbelts weren't working correctly for women. So that again, you know, that was not making development responsible. And that, you know, now we have these electric cars, the Teslas, these new electric cars, they're also made, you know, the way that your eye looks, basically the height is more built, not, you know, to fit all size individuals. So depending on the field of the research, it may entitle then an analysis of gender, sex, or both. So basically, the research question that you have may not matter, but it may matter. So the point is, as long as you are aware that there may be a difference in what you're looking at. So what I'm gonna do now is show you a video, because it's a beautiful video and it's on cardiovascular diseases, which has made some progress lately because of the recognition that the disease, cardiovascular disease, has different symptoms in the women and men. And you know, physicians are slowly realizing this, but it's been taking a while. So, ok. Perché non parte? Well, before it went, lo sapete, perché prima ha funzionato, vabbè, possiamo anche... No. Va bene. Is there a way to look at the, you know, the history, Vabbè. Otherwise, you can look at it.
Oh, maybe la cronologia dicevi.
Bravo, quello.
Ritorna sul...
Che io... Eccolo, è andato. Perfect.
Meet Mira and Leo. They're both 55 years old and have chest pain. Leo goes straight to the hospital, where he's admitted for testing. Leo's a smoker and has a family history of heart disease. Mira doesn't go to the ER. Instead, she takes her heartburn medicine and eventually visits her family doctor when her chest pain worsens. Mira is healthy, but has been feeling stressed recently. Her doctor suspects she's having a panic attack and sends her home. Later that day, Mira collapses and is rushed to the hospital. Mira and Leo receive their diagnoses. Both have cardiovascular disease. We're the same in many ways, but when it comes to our health and well-being, differences matter. Sex and gender shape us inside and out, from cells to society. Cardiovascular disease is a leading cause of death for both men and women. Heart disease in women is often unrecognized, while men tend to develop the disease at a younger age. Why? The answers are complicated, but we can uncover them with better science. Whether you're studying cells, tissues, animals, or people, integrating sex and gender in your research is an opportunity for innovation and more rigorous science. An increasing number of scientists are accounting for sex and gender, and funding agencies around the world now require applicants to consider sex and gender in their research. But there's more to do. In Canada, 1 in 3 women die from cardiovascular disease, yet most preclinical studies are conducted on male mice. The causes behind the earlier onset of heart disease in men still aren't fully understood. When it comes to health research, sex and gender can be tough to tease apart, but they're different. Sex refers to biological attributes, including physical features, chromosomes, gene expression, hormones, and anatomy. Gender refers to socially constructed roles, behaviors, expressions, and identities. Think of Leo. If gender influences smoking behavior among men, and smoking raises the risk of heart disease, where does sex end and gender begin? Accounting for sex and gender in health research is about effective health promotion for Leo and more personalized health care for Mira. It's about shaping science to create a healthier world for men, women, boys, girls, and gender-diverse people. At the Canadian Institutes of Health Research, we have developed online training.
Okay, good. So cardiovascular disease was really one of the first diseases that brought the attention to this, and it's true. Women cardiovascular disease is now the #2 killer in women because many women do not realizing they're having cardiovascular disease. Even the way a physician determines whether a woman is having a heart attack or not, or whatever, cardiovascular disease is very different between the genders. So slowly this is changing a little bit because of the awareness. So, for example, back in 2010, 10 drugs were withdrawn from the market because of adverse health effects. And what happened is that 8 of them posed higher risk for women than for men because of these physiological differences. So imagine that a company makes a drug and that has an adverse health effect. It can actually be lethal to one sex. And so not only, of course, the detrimental effect of that, but also the billions of dollars that are just spent, wasted because of the wrong way the science was conducted. Now this is changing slowly. It's changing because funding agencies are asking to discuss this, whether in your experimental design you have included possibilities, the difference of whether there's a possibility of a difference among the sex and or genders. It's slowly changing, but not fast enough, and I'll show you. So this is a graph from 2011 looking at the proportion of research studies. You can see throughout disciplines. Clearly you can see the male dominates except for reproduction. But the worrisome part is, I don't know if you appreciate the gray here, the unspecified, it goes all the way here. How many studies do not specify the sex of the cells they're using or of the animals? And this is even true for cardiovascular disease. I've just shown you a video emphasizing the importance of the disease, the difference in men and women, yet look at how many studies, how journals do not specify, have published unspecified sex in the cells that they used. This is slowly changing, not fast enough. This is from neuroscience. You can see the unspecified sex pie is diminishing, which is good. But this really triggered me last July. Nature Communication published a work from Sabine Orte-Prigione, who's done an incredible amount of work in this area, and show how sex and gender, the consideration of these two variables are lacking in COVID-19 clinical studies. Can you imagine? We've heard now for 2 years, men become sick different, yet the clinical trials and the studies do not disaggregate data for the sexes. That is, tremendous if you think about it. So we're still lagging behind. It's difficult. Why is it difficult? Because these two variables continuously interact. So the way we behave, our biology impacts the way we behave. And this is a continuum throughout our life cycle, our lifetime. So one example is, for example, through epigenetic. When you're in your mom's womb, right, what your mom eats, what she drinks, whether she's stressed, is going to influence your cells. And that's going to be throughout your lifestyle. And so you have this continuous interdependency between sex and gender, which are very difficult to tease apart, but important to be aware of. That's also true in animal research. This was published from Sorge in Nature Methods a few years ago, showing that not only cage animals that we all use for experiments are sensitive to social dynamics, caging, diet, of course, room, whether the temperature of the room is the sound, but also in this case, the sex of the researcher. So the mice smell the hormones that the researcher emanates, and that's sex, and then reacts differently to pain, whether is a female or a male researchers handling them. So of course this has, raises the issue again of awareness. This was a slide that I took from a keynote address from David Page, who was the director of the Whitehead Institute, and this really caught my attention because he was clearly discussing how when we think of diseases, for example, autoimmune disease, which has a preference for boys, boys unfortunately get this disorder more than girls, the autism spectrum disorder. If we think of autoimmune disease, we know women get autoimmune disease more than men. But these diseases are not diseases that are due to defects or they're dependent on the gonads. So there's something else at the infinite level of each cell that is could be perhaps responsible for this. So when we think of our cells, we have to think that in a male individual, every single cell is going to be an XY cells. And in a female individual, every single cell is going to be an XX cells. And so they may not matter, but it may matter. But in order to really understand at the finite level the molecular and biochemical difference between these two cells is of course of utmost importance. And it's important in the field of medicine to understand how the biology of these two cells, how and where do they differ. And this then has importance in science. This leads me to this concept of gendered innovation that was co-created by Londa Shibiber, and this was, for example, in pain research. We know that men and women, or females and males, feel pain in a different way. And this was published in Nature a few years ago, that in fact, in mice, the pain sensor for intermediate is different in males and in females. You have a microglia in male cells and you have a T cell in female cells. So of course that has tremendous impact, right, on pain, on the way pills may be developed that hit the two sexes in a different way. And for us, we work in cancer research with the BRCA. So, you know, the BRCA genes, the family, they became sort of more famous with Angelina Jolie having this disease. And we all know that, you know, BRCA, if a woman has BRCA, the mutation, she may have a greater chance of developing breast cancer and or ovarian cancer. So that's what we thought. In fact, it was called the breast cancer syndrome. But what has turned out lately is that also men that have these BRCA mutations, so if you have a BRCA mutation running in the family, men also need to be tested because they may have an increased chance of developing prostate cancer and or pancreatic cancer. So in Yale Hospital, about 40% of the patients that have prostate cancers, in fact, have BRCA disease. So again, you know, you apply a sex lens in a research project, don't only think of the way the sex and the gender behave, that will lead you to improve the health of both men and women. And one brief on COVID, you know, we've learned a lot about it, and clearly from the beginning, what we knew is that COVID was hitting the two sexes differently. Okay, so sorry, the infection rate of males and men and women is the same, but the virus is more lethal. The case fatality rate in males is higher. That's throughout the world and at all age groups. So when you think there's a sex dimension to the virus, because the way the virus enters the cells, the way the cell responds in all these different ways, so through the receptor, through the innate immune response, adaptive immune response, these are all regulated by sex genes and sex hormones. And of course, there's a gender dimension to it because, you know, we know that smokers are more at risk of developing severe COVID, and of course, smoking is higher in men worldwide. The preventive measures are different in the two genders. The occupation, women make up a majority of healthcare workers. And then, of course, the response to treatment is different in the genders. So this is the perfect example of a virus with a clear sex and gender dimension to it. And then this brings me just to the last point. With COVID, this other term, you know, beyond sex and gender is emerging, that, you know, we have to also think of this intersectionality. So when we think of science, we should not only think of these two variables, but also of all these education, ethnicity, race, religion, and how the COVID-19 has really brought out these inequities in our health system and societies. And this was taken again from the Canadian Institute of Health, where they really clearly, depict the importance of this rainbow in science. And when I think of it, I always think that us as in the scientific and medical community, we need to acknowledge this. By acknowledging this, that will also make our science and our research better. Again, an example is the bias that we have in medical devices. The famous oximeter, that we use to sense, that has a physical bias. It's the way it senses dark skin is different from the way it senses light skin. Of course, what that does, it leads to a computational bias, which leads to an interpretational bias. So, and this is true also for the human-machine interaction. There's an incredible bias in these. And so in the future, what people that work, engineers that work in this area, they should really include diverse subjects in the way to benefit all people. This, by the way, was just published in Science last month. One last point, again, Londa Shibiber really made us reflect on how, for example, genome-wide association study, 80% of participants are of European descent, which means that when you look at risk predictors, then you're failing, for example, if you want to look at patients from African ancestry. So in fact, they're more wrongly diagnosed with increased genetic risk of developing cardiomyopathy because the predictors have not been validated. So we need to think and make artificial intelligence that it's fair for all, which will bring us to the initial of the inclusive, to make clinical trials more inclusive. That was the beginning, right? The reason why a lot of drugs were made the wrong way because not enough women were included in the trials. That's why it was lethal in women. But of course, the reasoning behind that was good. Back in the 90s, they wanted to protect women. Perhaps, if a woman was pregnant and she didn't know, you didn't want her to expose to medicine in which you didn't know what the effects were. But at the end, what happened is the outcome was wrong. So now, trials are being made more inclusive, but not inclusive enough, because results are suggesting that more women and older people should be included in trials. So you can immediately see how these biases are, and that's going to affect us, each one of us. So finally, just to close up, we've built a teaching module that's up online. If you take this, you will learn how to integrate all these variables in your science. It's a website, password, it's free. It gives you a certificate. And I'd like to acknowledge all the people from Libra that worked. My lab, who is very patient with me. Of course, the head and neck division, I work with head and neck cancers. And finally, you for your attention. And thank you, Ferdinando.
Questions? So, I have a couple of questions. No, okay. So.
Well, thank you very much for this very clear description of the problem. Now I think probably, I don't know if I missed something, but there are clear possibilities to cope with this problem at the clinical level. Now I wonder how we that are more in the lab can cope with this with cell lines. We should throw them in the vessel somewhere?
No. I think the point of
it's provocative, it's just to discuss about this.
No, because if you think about it, right, I mean, I think by looking at the past, right, we learn and then we project in the future. So you know the whole story about the HeLa cells, that has a lot of implications. But you're not going to throw them away, correct? We all work on them. I think the point is to raise awareness. To raise awareness, for students, postdocs, even us, PIs, is when, you're doing an experiment, you say, I can ask you, so did you do an experiment yesterday in the lab in cells? Which cells did you use?
Which cell?
Che cellule hai usato? E che sono maschi o femmine?
Nobody checked
Exactly.
the sex
So the
chromosome.
thing is, just be aware, right? You start raising awareness that you're doing this, and of course, transform cells, it doesn't really matter, but it starts changing your mind. Changes take many, many years. To change, you need 20 years, 30 years. Think of the HPV vaccine, how it was developed now, So just be aware, you sign your lab book, you say, okay, I did this in HeLa cells. Well, everybody knows HeLa cells, but whatever cells, and just put XX or XY, because sex is either that or that. So awareness.
Yes, in fact, also I'm studying BRCA2, and so I got exactly the problem. When I talk about this, sometimes we also introduce in a not good way our talk, because we say, well, also BRCA2 may give some problems, more chemoresistance, but it's like a brackets in the prostate cancer. That's not the way we have to say. We have to say that BRCA2 is like P53, is an oncosuppressor, that's it.
In men and women.
In men and women. No, I got the problem. Thank you.
More questions?
It's always good
okay.
when you get questions.
Just an opinion about how much is important to discuss this gender mice or cell lines in grant, when you write a grant.
Okay, so grant funding agency are asking for it, not all of them. Italians, not very much, but all European Commission, NIH, most German funding agency, Canada, Ireland, they'll ask you, did you consider whether your research problem has a sex and gender dimension? If you work on prostate cancer, you're going to say no, of course. But you say, yes, I've considered it. However, my problem deals with, you just explain why, your model, it won't be important, because they want to make sure you get public funding. They want to make sure that their money is spent well. And again, it may not matter, but it may matter, but it does raise awareness. And I think that's the point. And the other point is now journals are asking for it. Major journals are asking, once your paper is accepted, they're going to start asking you the sex of the cell lines that you've used. So it's important just to know. I mean, you can find this information. And that brings you the matter of reproducibility. Sometimes you may not be able to reproduce a result, maybe because of the cell line that you use. It could be something that's sex dependent, although, in transformed cells, it's more difficult. But primary cells, a lot of us work with primary explants, and I'm sure not everybody knows whether they've been taken from a male or a female animal, so it's important.
Thank you for your interesting talk, and I think that in the future there will be a lot to speak about. I wonder whether may arise some conflict when we are dealing with preclinical model, so with mice, because we must keep in mind the 3R principle, and I think that there is a little bit of conflict between the 3R principle, so to reduce the number of animals, and the inclusive needs to have both sex.
Yeah, so
of course, if include females, we have a lot of bias coming from estrogenic cycle and many other things. So I think that there will be something to consider in the future.
So I didn't show you all the data, you know, so in our teaching module, we also discussed this. Of course, the 3R principle is a major principle, you know, you want to make sure that you use the mice in the correct way. So, of course, you know, in the case studies that we conducted, we asked scientists, and they say, well, of course, you know, it would cost less, and you use less mice if you run an experiment. But then you have an underpowered experiment, which, I remember one of them confronted it with a three, you know, running with a big car or a three-wheel bag. You know, at the end, the results are not going to be. Nevertheless, there's a paper by Natasha, I can't remember her last name, but it's on our teaching module, where she clearly discusses the way you can do statistics and still run experiments with enough statistical power using less mice and introducing both sexes. So basically you will not go, because this is a big problem, you brought it out. This is a major issue, the 3R and the ethical issue, of course.
We need also to change the mind of our reviewer at the Istituto Superiore di Sanità, because the big problem is how many animals do you will use.
Yeah, so I think with these statistical analyses, this is a paper that was published in Nature Communication just maybe 2 years ago, and there's a few more that have come out that really show you how, just by using 6 animals, how you can put both sexes in by using 6 animals and getting the same statistical powered experiment. So that's published. I don't work with animals.
I'm not sure that
yeah.
standard deviation remains the same.
So just one consideration. Clearly, when we talk about, for instance, cancer cells, so you show us that there are the new, for instance, breast tumors in men. But all of us, we work with the cell line, cancer cell line, stable cancer line, very, very old.
Yeah.
They are all female, okay? So now you mean that we need to find again these cell lines, because we don't have, usually. We have to buy, or you need an hospital.
No, I think you just need to be aware. You know, as I said, you just be aware that those cell lines are that. And of course, you know, once you start passenger cells, I mean, transform cell lines. But it's the whole idea that needs, it's the whole way of looking at things. And it may not matter, but it may matter.
And just another point. You say at the end that we need to include more older people in the trial.
Clinical trials.
Clinical trial. But this means that the pharmaceutical companies needs to change mind.
Exactly.
Because they prefer to have young people.
It's easier.
Okay. My point is, this gender and sex dimension is important for pharmaceutical companies, so they do what they want, anyhow. So that's the point.
Yeah.
Because it's better don't use okay. So.
But you see, I mean, if you think we can even discuss the COVID vaccine. You know, the side effects for the AstraZeneca for the women were much worse. The data is there. So.
Okay, thank you again to Susanna Chiocca for the talk. So I think it's time to move to the next section. Please, the chairs, they have to come here. So we are a little bit late, but okay, we can... Yeah, it's not your fault. No, absolutely.
Okay, we can start with the second section of this workshop concerning metabolism and epigenetic interplay in cancer. And the first talk is of Luigi Ippolito from University of Firenze. And the title is Lactate Rewires Lipid Metabolism and Sustains a Metabolic Epigenetic Axis in Prostate Cancer. Please.
like to thank the organizer for giving me the opportunity to be here in front of you after 2 years of pandemic. Thanks. To be here in front of you after 2-year pandemics to share our data about prostate cancer metabolism and epigenetics. Tumor strongly depends on their surrounding microenvironment, since trauma elements are required to support cancer cell proliferation, invasion, and possibly metastasis. To do that, metabolic dependencies and metabolic crosstalk between stromal and cancer cells have been established and identified during the last decades. Particularly, one of the most relevant nutrients within the tumor microenvironment is the lactate, which is secreted by glycolytic cancer and non-cancer cells and exploited within the tumor progression for multiple purposes. The expertise of our lab has provided evidence of a cancer-associated fibroblast and prostate cancer crosstalk. We refer to the isolation of primary patient-matched prostate fibroblasts from hyperplasia or benign tissue. We're referring to the healthy fibroblasts. And the cancer-associated fibroblasts were isolated from tumor areas derived from prostate cancer tissues. We have provided evidence that the cancer-associated fibroblasts derived lactate is able to shape mitochondrial changes in prostate cancer cells and enhance invasive abilities of these malignant cells. To have a more clear scenario of the metabolism in prostate carcinoma, we subjected fresh prostate cancer tissue to metabolomics analysis. As expected, we find higher levels of lactate compared to the benign tissue. But we also observe that citrate, lipid species such as palmitate, stearate, cholesterols are strongly enriched in carcinoma tissues, suggesting that these alterations in lipid metabolism are probably linked to lactate metabolism indeed. So, to assess this role, we analyze in a prostate tissue cancer assay the expression of MCT1, which is the most important lactate transporter for importing lactate within the cells. And as you can see, we find that the MCT1 expression are increased along the tumor progression. This also results in higher olive red lipid staining in carcinoma comparing the benign tissue, suggesting that high levels of lactate and lipids are correlated in prostate carcinoma. This also is confirmed by in silico analysis by publicly available prostate cancer patient dataset, indicating that MCT1 and other lipid pathway genes are positively correlated. So we skip to in vitro model, and we expose prostate cancer cells to healthy fibroblasts and CAF conditioning media and lactate to mimic CAF metabolic conditioning. And we subjected these cells to metabolomics analysis, and as expected, we find that most of TCI cycle intermediates are strongly incorporating lactate-derived carbons. But we focus our attention on the citrate because it is the first reaction step driver of lipid biosynthesis, and we also observe that indeed the palmitate, which is the first fatty acid synthesized upon the lipid de novo biogenesis, is strongly enriched in lactate carbons, suggesting that stromal lactate fuels lipid biosynthesis via citrate in cancer cells. To confirm this data, we also observe a significant enrichment of radiolabeled lactate in CAF and lactate-exposed cancer cells, and MCT1 inhibition prevents this accumulation. Also, we observe the expression of main lipogenic enzymes, such as fatty acid synthase and the activating the phosphorylation of acetyl-CoA carboxylase, which is revealed increase in CAF and lactate-exposed cancer cells. And also, the cholesterol levels are even induced by lactate in this context. Newly synthesized lipids can be stored in specific organelles the so-called lipid droplets, which contains triglyceride and cholesterol esters. As you can see, we can appreciate that calf and lactate-exposed cancer cells exhibit a higher enrichment and higher content of lipid droplets, as you can see by the body pie neutral lipid staining. And interestingly, the targeting of lactate entry and the lipogenic and cholesterol pathways are detrimental for the formation of such organelles. But which is the fate of the lipids derived from lipid droplets? It's already known that lipid droplets can undergo breakdown through the action of different lipases, such as ATGL, which drives the first reaction of the breakdown of lipid droplets themselves, in order to sustain fatty acid oxidation in the mitochondria. Seahorse analysis reveals that calf and lactate-exposed cancer cells exhibit a respiratory phenotype. But interestingly, if we treat our cells with ATGL statin, which block these lipases, so the breakdown of lipid droplets, this respiratory phenotype is decreased, suggesting that lipid droplets can be mobilized to sustain the mitochondrial activity of these cancer cells. But the metabolic activity of mitochondria in these cancer cells can be sustained by acetyl-CoA levels, but this molecule is quite important because it can be required into the nucleus to sustain histone acetylation and consequently gene transcription. In keeping, we find a higher level of acetyl-CoA in calf and lactate-exposed cancer cells, and again, the MCT1 inhibition prevents such accumulation. Interestingly, we observe that histone acetylation, particularly on the lysine 9 and 27 of histonase 3, which are the main markers of active gene transcription, are increased upon calf and lactate conditioning, suggesting that lactate drives histone acetylation via lipid rewiring. But the histone acetylation can be read, can trigger their signal through the action of different molecules. So one of the most important are the bromo domain extraterminal family protein, the so-called BET proteins, which are recruited to the acetylated histones. During the last decades, bromo domain inhibitors have been developed in order to block the transcriptional activation of cancer cells and other pathologies. And for this, we interrogated our experimental model with the BET inhibitor 76.2, and we observed a significant reduction of lipid species induced by lactate upon the administration of this drug. Interestingly, we also find that BET inhibition impairs the lactate-dependent lipid accumulation in lactate-exposed cancer cells, and this is also confirmed by using silencing for BRD4, which is one of the most important BET protein. At this point, we perform our RNA sequencing on lactate-exposed cancer cells with or without BET inhibition to discriminate which are the genes dependent on the lactate exposure and transcribed and activated via histone acetylation. Among the regulated genes, we focus our attention on this gene, which is PLIN2, because it is important in our model as it associates with lipid droplets and stabilize and enhance lipid droplets function. We appreciate that calf and lactate-exposed cancer cells exhibit higher expression of perilipin-2, and MCT1 inhibition prevents this expression. And we genetically targeted perilipin-2 in our model, and we observe a strong reduction in histone acetylation induced by lactate. This correlates with also degrees of invasive abilities of cancer cells induced by lactate and calf reconditioning. This correlates also with higher levels of perilipin-2 in a prostate cancer tissue microarray, showing that the higher graded prostate cancer exhibit higher expression of perilipin-2 compared to the benign tissue and the lower graded ones, suggesting that perilipin-2 is very important for lactate metabolism, for lactate-induced malignancy in cancer cells. At the end, we corroborate our in vitro data by a spontaneous metastatic assay. We co-inject prostate cancer cells with healthy and calf in nude mice. And we also have a cohort of animals supplemented with lactate on prostate carcinoma-bearing tumors. And then also we treat a cohort of mice with BET inhibition. As you can see by the lung section of the animals, we observe a strong increase of metastatic incidence of the derived from calf and lactate-derived tumors. And importantly, BET inhibition is able to decrease this metastatic behavior. Similarly, we use also perlipin-2 silence prostate cancer cells, and we observe similar results. And this suggests that the lactate-dependent lipid and histone acetylation axis is crucial for in vitro metastatic outgrowth. At the end, we can conclude that lactate provided by calf is used for multiple purposes. One of the most important can be the lipid anabolism in prostate cancer cells. And the lipid anabolism culminates in the formation of lipid droplets, which can be mobilized to support prostate cancer metabolism, but not only metabolism, also epigenetics. And we identify perlipin-2 as one of the most important lactate-dependent gene because it acts as a bridge between metabolic and epigenetic axis in these cancer cells. And is also crucial for sustaining the metastatization of prostate cancer. All these data are also have been published recently, a couple of weeks ago. I would like to thank the organizer and people of our lab, particularly Giusy Comito, who shared the first co-authorship of the work, Elisa Giannoni, Paola Chiaruggi, and all the rest of Andrea Morandi and Chiaruggi Lab, and you for attention.
Okay, thank you for the interesting talk, and we can start with any questions from the audience. Any questions?
I have one question. So at the end, you conclude saying that lactate is in some ways regulating this gene. But the main point is not lactate, it's the metabolic rewiring induced by lactate, because you don't have an idea, because essentially you don't know what are the transcriptional factors that are downstream lactate. So you mean, so because you see this upregulation, but do you have an idea what are the transcriptional factors that are activated by this metabolic rewiring, lactate dependent?
Okay, good point. Yes, I mean the metabolic rewiring induced by lactate metabolism, but I don't know exactly which are the transcription factors induced by the entry of the lactate. We focus on the metabolic rewiring and also the RNA sequencing promotes different activation and regulation of genes, and we focus only on the lipid-related genes induced by lactate. But which the major transcription factors we don't focus our attention on.
Because essentially what you see that there is an increase of acetylation, means that you have more acetyl-CoA.
Yes.
So in other terms, if you give to these cells acetyl-CoA, you will see the same panel of genes that are activated, or is it something that is quite specific to lactates? That is a second, because otherwise
I think it's quite specific to lactate. We performed some experiment with acetate, and we didn't resemble these the same results. Yes. So it is specific for the lactate for the induction of acetyl-CoA.
Yeah, that's the point. See, it means because it's not an increase of the acetylation that is quite in a specific way. But you have some specific genes that are upregulated or downregulated. I wait
yes.
for but you don't have an idea if there is a specific enrichment of a specific pathway or specific in your RNA-seq analysis.
RNA-seq cholesterol biosynthesis is positively correlated with the lactate. Is one of the genes that
there is a typical transcriptional factor that answers to the lactate. Okay.
No, at the moment, the transcription factors, we don't know.
Thank you.
My question is somehow related to this one, but I would like to understand why you, or I missed something in the talk, why you say that it's just specifically lactate from cancer-associated fibroblast. Anyway, because lactate anyway is a product of the aerobic glycolysis. So I would like to understand why the specificity of CAF originated.
Okay, because we perform metabolomics analysis on conditioning media derived from cancer-associated fibroblasts. We found that lactate is quite very enriched compared to other substrates that can be arise acetyl-CoA, for example, pyruvate or fatty acids. So we focus our attention on the lactate as the major substrate which are preferred by the cancer cells to be rewired and to be...
Okay, no, I mean, why you say that lactate is produced by cancer-associated fibroblasts?
Because cancer-associated...
But PCA cells do not produce lactate?
Yes, they produce, but only when they are alone. If you put in contact cancer-associated fibroblasts and cancer cells, they do not produce enough lactate levels, and they prefer exogenous
so
lactate.
You cannot rule out that there could be also some other molecule that you miss that can activate all this stuff. No, I mean, it's just because lactate is not specifically of cancer associated.
Yeah.
I don't know if I'm wrong, but...
No. But cancer-associated fibroblasts are strongly glycolytic cancer cells because they are even rewired by cancer cells contact through the action of some molecules, some cytokine, to be higher glycolytic compared to the healthy fibroblasts, for example.
Okay, thank you.
We proceed with the second topics from Giulia Robusti from European Institute of Milano. The topic is entitled Integrative Analysis of Aberrant Epigenetic Features in Triple Negative Breast Cancer Clinical Samples. Thank you very much.
Good morning, everyone. I'm Giulia Robusti, and I work in the lab of Tiziana Bonaldi at the European Institute of Oncology. And the project I'm working on is based on the integrative analysis of aberrant epigenetic features in breast cancer clinical samples with the focus on triple-negative breast cancer. Epigenetics has been shown to play a role in cancer, and in particular, histone PTMs have been described as general hallmark of cancers and as prognostic and diagnostic factors. And as well, histone-modifying enzymes have been described to be altered in several cancer types and to be potential therapeutic targets. Mass spectrometry has become the method of choice for the analysis of histone PTMs compared to other antibody-based methods, since it is unbiased, comprehensive, and quantitative, and it allows detections of combinations of modifications without a priori knowledge of the modification site. In our lab, we developed a series of methods for extraction of histones from clinical samples that we analyze with the introduction of an internal standard by gel digestion followed by liquid chromatography tandem mass spectrometry analysis. We have applied the mass spectrometry analysis to breast cancer clinical samples. Breast cancer has been described depending on different molecular features. In particular, 4 different subtypes have been so far described, and triple negative is the only one for which no specific markers have been identified so far. It is also a cancer characterized by poor outcome and by higher proliferation rate. It is also the most aggressive one. However, there are no specific targeted therapies available, and it is therefore important to select potential novel therapeutic targets. By mass spectrometry, we profiled histone PTMs in older breast cancer, in several breast cancer subtypes and normal tissues. And we were able to detect in a single run multiple histone PTMs for histone H3 and histone H4. And in particular, for the triple negative, we identified an epigenetic signature characterized by an increase of dimethylation on lysine 4 of histone H3 and K9 trimethylation of histone H3, a decrease of K27 trimethylation of histone H3 and K79 monomethylation of histone H3, and as well an important decrease of K20 trimethylation of histone H4 opposite to the increase of the K20 unmodified. So, in order to dissect the molecular and investigate the molecular mechanism underlying these aberrant epigenetic features, we are taking two different experimental strategies. One is the analysis of upstream mechanism, in particular to see correlation with altered histone-modifying enzymes. And the second is the analysis and investigation of downstream mechanism by looking at genomic distribution and as well as effect at the transcriptomic and proteomic level. And I will talk mainly about this part. We have decided to focus mainly on the increase of the dimethylation of lysine 4 of histone H3, in particular since it is changing mainly between triple negative and all the other subtypes. And from literature, we know that it is localized at promoter regions and associated with active transcription. It also correlates with gene expression. We selected a set of clinical fresh frozen samples, in particular luminal and triple negative, since these are the two subtypes that express the most divergent features and also express more different levels of K4 dimethylation. And we performed a multiomic analysis, including analysis of histone PTMs, ChIP-seq, transcriptomic, and proteomic analysis. By ChIP-seq analysis, we looked at the genomic distribution of the K4 dimethylation of histone H3, and as expected, we found most of common peaks between the two subtypes to be present in promoter regions that you see here in red. And interestingly, when we were looking at triple negative only and luminal only a few percentage of peaks were present in the promoter regions, while most of the peaks were in intronic and intergenic regions. Moreover, some of these peaks were also overlapping with the super enhancers, and I will talk about this. So we first look at genes, at peaks in promoter regions of coding and non-coding genes, and then at peaks in intronic regions of coding and non-coding genes. So we then integrated the data with RNA-seq data from the same samples, and in particular, we identified a correlation with expression. So as you can see here, when we have the peak in promoter regions of coding genes of triple negative only, we find an increase of expression. The same with luminal, in the case of luminal only, while no significant change is seen when the peaks are present in both subtypes. And a similar trend is present when we look at triple negative only genes. Then, for a more explorative analysis, we also looked at peaks present in intronic regions. And the trend was the same, so correlation with expression, in particular for the triple negative only. And then we also looked at peaks in intergenic regions. And here, as you can see, there is an increased expression compared to the peaks in the promoter regions. So we asked why this was happening, and we actually identified most of these peaks to overlap with super enhancers. So super enhancers are different from enhancers since they are clusters of enhancers far from the transcription start sites, And they have a higher effect at the transcription level, so they actually boost transcription. And this was very interesting for us. We looked in literature and we actually found that super enhancers seem to play a role, in particular in driving oncogenic expression in triple negative. And for an explorative analysis, we integrated our data with already published data from TCGA database and CPTAC, and we identified some interesting genes that seems to be upregulating depending on the presence of the peaks for decay for the methylation of histone H3. So we are now investigating these genes that could be possible, that would be interesting to study. We also look at peaks present for K4-demethylation of histone H3 to be present in non-coding genes, at promoters and intronic genes. And most of these genes were long non-coding RNAs. These were also described to be potential novel biomarkers in solid and therapeutic targets in solid tumors. And in particular, we found these two to be already described in the triple negative subtype, and also to have a role as in proliferation and to correlate with poorer prognosis. On the same samples, we also performed the proteomic analysis. In particular, we identified a total of 7,000 proteins, with 83 proteins up in triple negative and 105 up in luminal, with a p-value threshold of 0.01. And we did an integrative analysis by integrating all the data obtained. So we did a 2D enrichment annotation. And as you can see here, we can see the correlation between proteome and transcriptome. And by looking at positive correlation in triple negative, we observed that most of the terms were involved in immune and inflammatory response. In particular, we also filtered this data based on the presence of K4-demethylation of histone H3. So it was interesting to see that there is this immunological component, and actually the immune system seems to play an important role in breast cancer, and as well tumor infiltration, immune infiltration was described before in triple negative. So we are trying to study this point. And as well, the selection of possible candidate genes is ongoing, and we plan to manipulate this gene genetically and pharmacologically in model breast cancer cell lines. So with this, I would like to thank all the components of the TB group, in particular Tiziana and Roberta for supervising me in my PhD project, and Alessandro, who is the bioinformatician who did all the integrative analysis, the members of the mass spectrometry unit, the collaborators, and all of you for your attention.
Thank you very much, Giulia. Any question for audience?
Thank you very much. Nice talk. I have just one question because you have compared the methylation between luminal A and the triple negative, but do you compare also luminal B, or have you idea about the methylation pattern of luminal B? Because the luminal B is between the estrogen receptor positive breast cancer are more aggressive and show more resistance and are more similar to triple negative.
Yeah. So, of course, the focus of our study is the triple negative subtype, and we have decided to compare it mainly to the luminal because it's the one that is showing the opposite features, but also histone PTMs are changing between triple negative and luminal B subtypes. So we are also comparing to the other subtypes as well, the luminal B. Yeah, it was just a choice because our focus is mainly the triple negative, that is the one that is that has no specific features, so we are focusing on that, but we are comparing it also to other subtypes.
Because it could be interesting because for a problem, for example, for the resistance or for the prognosis. Thank you very much.
Okay, thank you.
I have a couple of questions. One, you say at the end that you have so an enrichment in immune response. It means that you have a target of the immune cells. So it means immune cells that activate in cancer cells a response. Mm-hmm. This means not that you have something that is of the lymphocyte, neutrophils, monocyte. So the genes that you see is something that is related to cancer cells.
Okay, so actually we don't exactly know which cells we are looking at because we are working with clinical samples, so they are very heterogeneous. So we don't know if the increase of the K4 dimethylation of histone H3 is dependent on the tumor cells or on the lymphocytes. So the next step will be to do immunohistochemistry for these markers that we identified. And it would be also interesting to do laser microdissections of these tissues to see if this depends on, for example, tumor infiltrating lymphocytes, or it depends on the tumor.
Okay, so it means that you have a mixture of the data.
Because we don't know which cells we are looking at. We usually use tumors with at least 60–90% of tumor cellularity, but we are not actually sure at which cells, which type of cells we are analyzing because of the heterogeneity of the tumor.
This is related to the second questions. If you think that in your proteomic data there is a bias for the high expressed proteins, this can in some way interfere with your analysis.
Yeah, we are thinking a bit about it, and in a certain way we are worried about it as well. But we usually select these tumors depending on the tumor component. So we are, let's say, sure that we are actually analyzing at least at proteomic level, the tumor cells.
Thank you.
Okay, thank you. And then let's start to the third presentation from Samantha Raboni from University of Parma, entitled "A key silencing histone marker on chromatin is lost when colorectal adenocarcinoma cells are depleted in methionine by methionine gamma lyase."
Good morning. Thank you for the opportunity to share the results of our research on the epigenetic changes induced by enzymatic methionine depletion on HT29. Among cancer metabolic signature, the elevated requirement for methionine is that is known as methionine dependence or Hoffman effect, represents a specific vulnerability, since the inability to survive to methionine deprivation distinguishes cancer cells from normal ones. Methionine is an essential amino acid crucial for cell growth and function due to its role in protein synthesis as a building block and in cellular metabolism as a precursor of S-adenosyl methionine. Methionine dependence and depletion has gained increased recognition as a tumor-specific therapeutic strategy, and Okay, and the bacterial methionine gamma lyase, a methionine degrading enzyme, represents an attractive drug candidate by targeting this metabolic weakness. Indeed, treatment with this pyridoxal phosphate-dependent homotetramer that converts L-methionine to alpha-ketobutyrate, ammonia, and methanethiol via gamma elimination reaction proved to exert anti-tumor effect in cancer cell culture and in a mouse model. Interestingly, FDA approval of asparaginase, a well-established asparagine depleting agent used to treat acute lymphocytic leukemia, demonstrated the power and applicability of nutrient starvation in cancer therapy. However, the detailed molecular mechanism explaining how restriction of methionine affects cancer cells still remain elusive. In eukaryotic cells, DNA wraps around an octamer of histone proteins, the basic repeating units of chromatin, and methyl and acetyl group are deposited on lysine and arginine, are the principal histone post-translational modification that regulate chromatin structure and gene expression in combination with DNA methylation. Since methionine starvation may perturb S-adenosine methionine availability and consequently the abundance of methyl group for histone methylation, we applied LCMS to accurately profile histone post-translational modification in response to MGL treatment for 24, 48, and 72 hours. The analysis of highly basic histone requires an optimized bottom-up proteomic approach in which propionylation of lysines generated tryptic peptides of optimal length, and propionylation at the N-termini increases hydrophobicity for optimal chromatographic separation. Cell vitality measured by trip and blue exclusion assay and metabolic activity evaluated by MTS assay together with microscopy observation monitored the progressive decrease in cell fitness that occurred during the time course of the histone profiling experiment. Oops, sorry. No, that's true. The methionine starvation did not alter the level of total histone methylation, but it did decrease the some specific histone marks. In particular, we found that H3K9 demethylated was decreased, and this is fundamental to maintain compaction on constitutive heterochromatin that is rich in transposable elements and microsatellite repeats. Together, also a depletion of H4K20 trimethylation that is associated with the repression of endogenous retroelements in literature was found. And treated cells also showed a decrease, a loss in H4K20 demethylation that tended to accumulate in association with the cell cycle progression. Treated cells also display an increased acetylation at position 14 and 23 of histone 3 that are mark of open chromatin, and we found a reduction in phosphorylation of serine 10 on histone 3 that is characteristic of non-actively dividing cells. Based on these results, we evaluated the expression of major satellites unit in treated cells, and the increase that we found in association with the reduction in the aforementioned repressive histone marks suggested that methionine-deprived cells may have impaired ability to keep silenced domains of DNA repeated elements. Evidence has been accumulating that it is important to maintain transcriptional control over the activation of transposable elements to sustain cancer progression, and that the absence of repressive histone marks may reactivate the expression of these transposable elements, leading to viral mimicry response and cell growth inhibition. When we checked the analysis of the proteomic response to methionine scarcity, we found that a lower overall content of proteins in treated cells compared to control, as can be shown by a larger distribution of proteins in the negative end of the volcano plot. Proteins depleted by the MGL treatment mostly belong to the RNA binding molecular function, and we found that downregulation of protein participating in RNA splicing, degradation, and transport made us think that cell may tend to minimize the production of protein involved in transcription to translation. Thus, in conclusion, we think that our research provides some hint of the events occurring to methionine-depleted cells upon MGL treatment. And by exploiting a combination of mass spectrometry, quantitative PCR, and cell culture assays, we demonstrated that methionine treatment causes cytotoxicity affecting chromatin and proteome regulation and stimulating spurious transcription in cancer cells. Based on these results, we are interested to further explore the link between MGL-mediated methionine deprivation and chromatin perturbation, and we would like to get to a deeper insight into the cellular and epigenetic mechanism involved in methionine dependence. Also, our research effort is aimed to define targeting and strategies and delivery strategies for MGL. Indeed, we are exploring the association with the molecules that specifically recognize determinants preferentially overexpressed on tumor cell surface and different conjugation approaches with the nanoparticles. We think that these strategies will improve the therapeutic efficacy of MGL and will foster a more effective clinical development of this enzymatic anti-tumor approach. So finally, I would like to acknowledge the contribution of all the people involved in this research, people from University of Parma and our collaborator in the United States that gave a very important contribution on helping us understanding the histone post-translational modification world. And I thank you, all of you, for your attention.
Okay, thank you for the great talk, and we can start with the discussion. Any questions? Yeah.
Okay, thank you. Nice talk. So I'm curious to understand if you used only this approach using an enzyme to remove essentially methionine from the medium, or you tried to inhibit the import of methionine in the cell, simply because we are working on other metabolites or other amino acids, and to understand if you compared, if you have something interesting to say about removal from the medium or inhibiting the import of the amino acids into the cells. Thank you.
Okay, that's an interesting question. The thing is that we checked the catalytic performance of MGL in the medium is pretty much fast. So when you add this enzyme to the growing medium for cells, methionine coming from the medium is the consumption of methionine is completed pretty much in 1 hour. So the slow effect that you see, because cytotoxicity is then obtained at pretty much 72 hours, that's the IC50 that we can calculate, means that when you remove the methionine, it's almost instantaneous, and then you start in the cell a lot of adaptive pathways. So I don't think that the transport would really be the case for our enzyme, but of course, because I think there will be something that rewires the salvage pathway of this enzyme and of the methyl group inside the cell to try to find a solution to the consumption that the enzyme produces. But of course, transporter for methionine are known, and we checked if there was some difference in the proteomic.
I'm just suggesting this because we have very surprising effect, not inhibiting 100% of the import, but you have, so in our case, clearly it can be different. But we have very striking effect only inhibiting like 30%, 40% of the import. You see important differences in the activity of the cells.
Okay.
So maybe it can be interesting to understand that. Anyway.
No. That's for that's always a very good point. We had another point of view, starting from the fact that the enzyme removes methionine pretty fast, and so we didn't think that the 2 time frame were on the same plan. But yeah, that's a good point, and we will think about it.
Thank you.
Thank you for the talk. Just a discussion on one point. Are you going to perform diet restriction in animal models for methionine? And if yes, what do you expect on the effect of this restriction on other cells, for example, T cells function?
We actually are working at this time, we're working on cell culture, so we are not moving to animal models soon, because that's not part of our expertise. Literature published either enzymatic starvation on animal models than diet deprived animal subjected to methionine starvation. Actually, I'm not really competent on the possibility of comparing the result of the two. The thing that has been pushing the research on on the enzymatic methionine depletion is that thinking on a translational step, dietary starvation is quite challenging, considering it as a therapeutic tool. So enzyme would be more like a drug and could be more easily thought as a clinical approach.
Okay, thanks.
No more question? Okay. Thank you very much. The last speaker is Marina Lachimia from University of Catanzaro. She will talk about novel enzyme 2DJ1, a key player in epigenetic misregulation in breast cancer.
Okay, good morning, everybody. First of all, I would like to thank the organizers for giving me the opportunity to present my recent data. So today I'm going to present to you my PhD project, which is focused on studying a particular protein called DJ1 as a key player in epigenetic misregulation in breast cancer. First of all, we know that one of the main hallmarks of cancer cells is the metabolic rewiring. And in particular, we know that the Warburg effect is one of the main features of breast cancer cells and consists into a switch toward anaerobic glycolysis, even in aerobic condition. The establishment of the Warburg effect in cancer cells leads to the establishment of a stress condition which is very damaging for the cells, which consists into a accumulation of reactive carbonyl species, which act as glycating agents and are able to react with lysine and arginine residue on proteins, giving start to a chain reaction that ends with the irreversible formation of advanced glycation end products. One of these reactive carbonyl species in particular, which is methylglyoxal, is able to enter the nucleus, where it can react with histone tails, leading to cell apoptosis due to a fatal alteration of the histone code. So, in theory, cancer cells should undergo apoptosis due to the establishment of carbonyl stress, but in reality, this doesn't happen because there are two known strategies that cancer cells can use to prevent this type of cell death. The first one is citrullynation, which is an enzymatic reaction catalyzed by PAD enzymes, which consists into the switch from arginine to citrulline in order to prevent glycation. In a previous work, we have already shown how HCC 1937 cells, which are cells characterized by high glycolytic activity, show higher levels of citrullination. The other strategy to prevent cell death induced by carbonyl stress is the overexpression of this protein called DJ1, which play lots of different roles into the cells. And for example, it is an important antioxidant. In this case, it plays an important role as a deglycase enzyme, and it can remove MGO adducts on the proteins in order to prevent cell apoptosis. So, for this reason, we think that this protein has a survival role in tumor cells, and so we have decided to study this protein using a proteomic approach. First of all, we started by searching for glycation sites on histones. So we performed a bidimensional tau electrophoresis, and then we identified the H-spot through mass spectrometry, focusing on methylglyoxal as post-translational modification. And we identified 17 different glycation sites. Then we performed a cellular metabolism analysis on two breast cancer models, which are MCF7, which is a model of sporadic breast cancer, and HCC 1937, which is a model of hereditary breast cancer. And as you can see from the graph of extracellular acidification rate, HCC 1937, which is the hereditary model, show the highest glycolytic activity. Then we performed some Western blot analysis, which confirmed that both DJ1 levels and MGO levels are higher in the cell type with the highest glycolytic activity. Then we performed a bidimensional electrophoresis of the whole extract from our cells, and we noticed that DJ1 in the bidimensional map doesn't appear as a single spot, but as a chain of spots, suggesting the existence of different proteoforms. So we identified them through mass spectrometry, and this allowed us to identify a novel phosphorylation on threonine 125, which is very interesting because for its proximity to the catalytic site, that suggesting a potential regulatory role of the glyoxylase activity. So we performed a scan site analysis, which suggested that the phosphopeptide, which contain the threonine 125, encloses a putative AKT consensus. This data was then confirmed performing an immunoprecipitation analysis of phospho-AKT substrate. Then, in order to better understand how AKT pathway can modulate DJ1 proteoforms, we performed an experiment both activating and inactivating the pathway. First of all, we inactivated the pathway using LY, which is a PI3K inhibitor. And the treatment, as you can see from the Western blot, induced a significant shift of DJ1 proteoform toward basic pH, according with the loss of the acidic group from phosphorylation, while the activation of the AKT pathway that we obtained treating cells with the H2O2 induced a shift of the proteoforms toward acid pH, according with the gain of the acidic group. Then, to better understand how the pathway is crucial for the modulation of DJ1 localization, we performed an immunofluorescence analysis, and both in normal condition and in oxidative stress condition. As you can see, in normal condition, the DJ1 localization is mainly mitochondrial, because DJ1 is a mitochondrial protein, while in oxidative stress condition, DJ1 translocates into the nucleus to perform its deglycase activity. Then we wanted to better understand how DJ1 interacts with nucleosomes, so we performed an immunoprecipitation assay using anti-DJ1 antibodies on the nuclear fraction, and then we identified the interactomes with mass spectrometry. We identified several proteins. The most interesting were histone isoforms. Then, since we thought that the phosphorylated isoform was the only one interacting with nucleosomes, we performed a bidimensional tau electrophoresis of histones. And in the bidimensional map, apart from the common histones spots, we identified another spot that was identified through mass spectrometry as phospho-DJ1. And this data was then confirmed by performing a Western blot, a bidimensional tau Western blot. The interesting thing is that in the Western blot from the whole extract, DJ1 appears as a train spot, while in the Western blot from the nucleosomes, DJ1 appears as a single spot. And that means that the only proteoform associating with the nucleosome is the phospho-DJ1. So this data confirmed that the AKT pathway is fundamental for the phosphorylation of DJ1 and for its glyoxylase activity. So we performed a last experiment where we treated cells with the H2O2, both in presence and in absence of LY. And as you can see, in every case, AKT inhibition is always followed by reduced levels of DJ1 and higher levels of MGO on histones. So that means that the phosphorylation performed by AKT is fundamental for glyoxylase activity. These data confirm how the DJ1 is a crucial protein for cells, for cancer cell survival, And for this reason, we are investigating it as a potential therapeutic target. And today, lots of work are being carried out in order to identify potential DJ1 inhibitors. In particular, we are trying to detect a possible inhibitor which target, in particular, the phosphorylated proteoform of DJ1 in order to preserve the antioxidant function of the non-phosphorylated proteoforms. So we have already started our research, and we are collaborating with the pharmaceutical chemistry group of our university in order to individuate this molecule. And here you can see the molecular modeling analysis of a potential DJ1 inhibitor, which can interact both with the normal DJ1 and with phospho-DJ1. So in conclusion, we have elucidated with our work, and we are still elucidating, a strategy used by cancer cells to preserve their immortality, disclosing a novel therapeutic target and pointing out the notion that the modulation of specific DJ1 function might produce substantial anti-cancer effects. And the final aim of our work at this moment is to verify the effect of phospho-DJ1 inhibition in association with other classic antitumoral drugs. So thanks for your attention, and thanks to my collaborators.
Thank you very much for your nice talk. Any question?
Oh, very interesting. I would like to understand if I understood correctly that DJ1 is a mitochondrial protein.
Yes.
Do you know physiologically when DJ1, is released by mitochondria, in which conditions, in order to relocalize into the nucleus?
We have proved that DJ1 is translocated into the nucleus when AKT pathway is activated. So in oxidative stress condition.
Oh, okay. But you don't know anything about mitochondrial function during...
the main function is an antioxidant function.
No, in which condition of the mitochondrial physiology, in which condition you have actually relocalization of these mitochondrial proteins to the nucleus? Only in oxidative stress conditions?
We have only seen it in oxidative stress condition.
So it is not known actually what is the triggering of this movement between mitochondrial and...
Not at the moment.
Okay.
We are still investigating it.
Thank you for your talk. My question is, it's quite, so this is my opinion, it's quite strange that you go to study an antioxidant protein by inducing AKT through H2O2 stimulation.
Mm-hmm.
So do you think it's better that you stimulate AKT with the serum, with what you want, serum starvation release? But when how you can exclude that the effect that you show is not, so it's the H2O2 that you add to your cells that has a secondary effect activate AKT. So do you think it's better? So probably if you use, for example, other cell models where there is oncogenic KRAS, hyperactivation, AKT mutant, constitutive active, you don't need really to put H2O2 to see your phenotypes. So this movement in the nucleus. So maybe it's better.
I never thought that.
Yeah, because it's an antioxidant, but you put H2O2 to activate AKT.
Yes.
And then you see the movement. Maybe it's better to
Maybe
avoid
this
oxidation
yes.
through H2O2. And the other point that was, so when you see this movement in the nucleus, doesn't mean that the mitochondria, there is no more DJ1. No. Still is in the mitochondria. See. Mm-hmm. So under stress, there is in the nucleus, DJ1, and the mitochondria, in both sides. Okay, thank you.
Thank you.
Okay, thank you. And we can go to the lunch, and we will restart to the third session at 3 p.m.
Please, one question. So, no, one question. Just one. Remind you, 3 o'clock, please. And second, what is important that the sponsor help us to do this. And some sponsors say, oh, it's strange, they escape away from our desk. That is not correct. So please go to the sponsor. That means that you have to stay in the queue, so there. But go to the sponsor, talk about the issues and so on. Thank you.